So good morning, all of you. So I'll begin my talk with which I've titled as understanding the initial steps of eukaryotic initiation, eukaryotic protein synthesis, and its regulation. So so if you remember the last talk by Professor Helmut, so he mentioned about molecular machines in the cell. And I would like to just bring to notice that this term of molecular machines for protein assemblies in the cell was first used by Bruce Alberts. And what he mentioned was that the entire cell can be viewed as a factory that contains an elaborate network of interlocking assemblies, each of which is composed of large set of protein machines. And the reason he termed it as machine is mentioned in the he later writes that these we called as machines precisely because, like machines, these protein assemblies contain highly coordinated moving parts. And ribosomes is one of such molecular machine that I'm going to talk about. It is if we go through the central dogma of life, which is like the DNA, which is transcribed into RNA, and this RNA is then translated into protein. So this function, job of translating the information which is there in the genetic code in the RNA to a protein is done by ribosomes. Now, in protein synthesis, we can briefly divide this into three steps. And so initiation, elongation, and termination. Initiation is nothing but the assembly of ribosomes at the start codon. If we go stepwise, then the small subunit of the ribosome binds at the start codon, followed by the recognition of the start codon by a specialized tRNA called as the initiator tRNA, and subsequent the large subunit binds to form the elongation competent ribosome. So initiation is the stepwise assembly of ribosomes at the start codon. If we look at the initiation pathways across the organisms, we find that we have, one is the canonical pathway, which normally takes place, and then there are certain other non-canonical pathways. I'll be talking about one of the canonical pathways in eukaryotes. And if we look through the three kingdoms of life, we see that in bacteria, the initiation pathway is the simplest in terms of the number of factors involved. So it involves only three factors, and there are, three factors are made up of just three polypeptides. In archaea, we see that there are a certain more number of factors involved, and there are factors which are even multi-protein factors. So hence we have 7 polypeptides. In eukaryote, it is much more complex, and we see 8 initiation factors. And some of these factors are again multi-protein complexes, and hence we see 18 to 25 polypeptides involved. So I'll briefly go through this, explain the eukaryotic translation initiation with using a schematic. So if we look at this small subunit, which is 40s, the 40s can be divided into two parts. Roughly, this is the head of the 40s and the body. And this A, P, and E are the three sites where the tRNA binds. So A site is the position where the amino acid tRNA binds. P site is actually the site of action where the initiated tRNA comes and binds. And E site is where the exit site. So after the peptide, the transfer of amino acid to the growing peptides, the empty tRNA moves to the E site, and from there it exits the ribosome. Now, if we go through the initiation pathway, one of the first steps in initiation is the formation of a complex which is 40S1, 1A, and EIF3. 1 and 1A, EIF1A binds at the A site, whereas EIF1 binds at the P site. So these are small initiation factors in the range of 10 to 15 kilodalton. Whereas EIF3 is a multi-protein complex. In Saccharomyces cerevisiae, it is formed of 6 subunits, whereas in mammals, we have up to 13 subunits. And hence their molecular weight ranges from 400 to 800 kilodalton, and they bind at the solvent side of the ribosome. The formation of this 40S11A sets the stage for the binding of the tRNA, and tRNA in this case is recruited by another protein complex which is called the EF2 complex, and EF2 is again made up of 3 different subunits: alpha, beta, and gamma, so roughly about 150 kilodalton, and often another factor called EF5 is recruited along with EF2. EF5 is a gap protein which actually enhances the GTPase action of in the EF2. Following this, the mRNA is recruited. In eukaryotes, the mRNA is capped, and hence the 5' region has a cap, and this is recruited again in complex with another protein complex, which is EF4 complex. And EF4 again has 4 subunits: EF4E, 4G, 4A, which is a helicase, and 4B. E is a cap binding protein, 4G is a big, large scaffold protein, A is a helicase, and B is an helicase activating protein. And together, this complex is again in the range of more than 200 kilodalton. So now the mRNA is bound at the ribosome binds to the mRNA at the 5' end of the RNA, and then it scans the untranslated region. So here I am showing the scanning of the ribosome of the untranslated region. So here you can see UUU codon, which is there. And so it scans this untranslated region to actually search for the start codon, which is here, which is AUG. So one of the complex that we see is a scanning complex, and in this conformation is called as a scanning conformation, or a POUT conformation, or an open conformation of the ribosome. Once the start codon is recognized, so now this has scanned and approached, reached the start codon, then it undergoes certain changes. One of the major changes that the EF1 is released from the complex, and then the subsequent factor downstream events occur. So this stage or this conformation of the ribosome is termed as P-in conformation, or the scanning arrested conformation, or closed conformation. So once the start codon is recognized, it achieves this conformation, EF1 leaves, and dissociation of EF1 then triggers the further downstream steps, which I'm not showing, so which would lead to the release of almost all of the factors except EF1A. And then another factor called EF5B joins the system, and it forms the elongation competent ATS. So before I go further to my talk, I'll just briefly explain, the method that we have used to determine these structures, which is cryo-electron microscopy. In cryo-electron microscopy, we use a very small amount of sample. So one has to prepare the sample, which could be a protein of interest, or a multi-protein complex, or in this case, ribosome protein complexes. Now this sample is then laid onto an EM grid. So this is a metallic grid, and each box within this grid has a pattern of holy carbon. So what we do is we apply the sample onto the grid and blot it with the excess of sample with a filter paper, and then this is plunge frozen in liquid ethane. And by doing this, what we are able to do is we are able to capture a given macromolecule in multiple orientations, and this is fixed. Now, when it is viewed in under a microscope, electron microscope, what we image is the different views of the macromolecules. So here you are seeing what may appear as black dots are actually different views of the ribosomal subunit. So multiple images are collected in tunes of thousands of images are collected, and these particles are picked up. Now they are computationally, there are softwares available which can now align these views. So similar views are aligned together in a process called 2D class averaging, and then all these multiple views are then combined together to give a final map. Now, when we began this work, so I'll begin with the work that I did in Professor Venki Ramakrishna's lab in Cambridge, LMB Cambridge, and then I'll continue to what we are doing now. when we started looking at this, we initially wanted to check at what is the conformation of the ribosome, or the conformation of the ribosome at which it is recognizing the codon.
I think there's a question.
Yes.
Yeah. Uh, Someone raised their hand. Can you go ahead and ask your question? Unmute yourself and ask.
Can you hear me?
Yeah, we can hear you. Can you increase your volume a little bit, please?
Okay. Is it okay now?
Yeah, it's better. Go ahead.
So can you please go back to the previous slide?
Yes.
So my question was, is it a single snapshot that you are showing, this micrograph picture? That
Yes, this is one micrograph picture only here.
this is at one time you are finding these different orientations.
Yes. In one time we find, because when we capture them, when we plunge freeze them, so they are randomly there first in the buffer. So as soon as you plunge freeze, it will get captured in whatever orientation they are. So yeah, so we are able to see multiple orientation, and that really help us to combine it into to form it into a 3D image. And if we are able to get multiple conformations, then then there's a bottleneck, and we are not able to come to the final map.
Thank you.
Okay. So the strategy was to capture it at a point where it is recognizing the start codon, and then we use certain biochemical strategies to do that. I'll just briefly mention that we used a simple E system because it's a much simpler system, and then we can to do so we can use the mRNA, which is without a cap, and hence we do not need EF4 and EF3, which are big assemblies, to do that. Then we used a fungi where we can work in actually lower pH, and that was to minimize the deacylation of the tRNA. Then we made use of certain mutations which would actually favor this particular conformation. So we went ahead and solved the structure, and when what I would like to mention here is that now one can actually do what we call as 3D classification. So where the data can be classified based on different classes, and here the algorithm divides data in such a way that it will divide the class based on the densities, different densities, or presence or absence of densities or things. So in by doing so, we were able to capture in one particular complex, which was EF1 and 1A and 1. So here we see that EF1A is bound at the A site, while EF1 is bound at the P site. And we were also able to capture the tRNA recognizing the start codon. So we can see now the mRNA. I'll come to the details a little later. The tRNA in green, EF2 alpha, and EF2 gamma, and beta. So beta, we just saw a proportion of it. So just going, so when we compare the two structures, what we see is that although the body of the ribosomes remain more or less the same, but the head rotates. So we see a large conformational change in the head of the ribosome. And the reason, the consequence of this particular rotation is that there are two RNA helices. One RNA helix moves in, whereas another RNA helix moves out of the P site. And this actually helps the moving out of this RNA helix, helix 31, actually allows the tRNA to accommodate deeper into the P site, and hence it is able to recognize the codons in the mRNA. The movement of this RNA helix into the P site brings in the conserved residues from the RNA helices, which then recognizes the GC base pair, which is present in only initiated tRNA, or mainly in the initiated tRNA. So it helps in the recognition of the initiated tRNA. Now, so we were able to see the codon, the anticodon of the tRNA recognizing the start codon. So here you can see the AUG. And if we look at in simple figure, what we noticed further was that the adjacent residue, that the -1 nucleotide of the mRNA and the +4 nucleotide, they were actually stacking with. In this case, an rRNA residue and the +4 with the tryptophan of EF1A. And by doing so, it would allow the mRNA to pause exactly with AUG at the P site, and and hence it would allow for the recognition of the AUG with the initiated tRNA. So this could explain or show us how the conformation of the ribosome while it is recognizing the start codon. Further, what we noticed was we were able to see the an N-terminal tail of EF1, which was not observed till date. So if we look at the sequence of N-terminal tail of EF1, we notice that it is number one, it is highly conserved across the organism, and we see a presence of three glycines in a span of 4 residues. It was known that this N-terminal tail is important for start codon recognition, but the details were not known. We were able to see the N-terminal tail in the map, and it approaches the codon-anticodon recognition such that it bends right exactly at the start codon. So, and this is where the three conserved glycines are present. So it helps to bend the loop near the codon-anticodon interaction. With EF1, with EF2 alpha, we were able to see the conformational changes. So if I compare the structure on the ribosome compared to structure of EF2 alpha with the tRNA away from the ribosome, we see that major conformational change where you see here domain 1 moves down. And this movement is essential to allow these arginines, these conserved arginines from 2 alpha to recognize the Kozak sequence in the mRNA. And Kozak sequence is an important sequence which provides context to the start codon. We were able to capture EF1. So what I had mentioned earlier in my introduction, that EF1 leaves the start codon, leaves the ribosomal complexes after the recognition of the start codon. Here we were able to capture the start codon, EF1 before it was released. And when we compare it with the this complex with the just 40S11A complex, we notice that EF1 has actually moved from its original position, and we see major changes in the movement of its two loops. So the original position would have steric clash with the tRNA, but in this position it is able to accommodate, and this movement actually allows the EF1 to accommodate initiated tRNA in the P site. So essentially what it tells is that the original position of EF1 would not allow initiated tRNA to accommodate completely into the P site, and hence only the recognition of the proper start codon AUG would then force EF1 away from out of its original position. So in some sense, EF1 promotes the fidelity of this start codon recognition by actually destabilizing the P state. And this is how it performs its function of recognizing the correct start codon in the mRNA. So we were able to solve this structure. However, this structure was without the component which is EIF3. So we solved further structures where we were able to see EIF3 both with the cognate AUG codon and also a non-cognate. I'll mention the reason for this doing this non-cognate, which is AUC. The reason to use a non-cognate codon was to capture it in a scanning conformation. So I'll just briefly mention that. So we again collected the data where we were able to now see EIF3 bound to the complex. So this is in pink is the component of EIF3. So it largely binds at the back of the ribosome. However, we were able to see some parts of the EF3 also on the subunit interface. As I mentioned earlier, we used an AUC codon to capture the ribosome in a scanning conformation. So we use certain other biochemical tricks, for example, to use a wild type tRNA and a wild type EF2 and avoid EF5 because EF5 shifts the equilibrium towards the PIN state, which is a scanning arrested state. And we also use recombinant EF3 so that we are sure that there is no EF3 contamination, because the EF5 often co-purifies with EF3. So by doing so, we were able to capture a scanning conformation of the ribosome. And if we look at this particular map, again, we saw very similar complexes where EF3 is bound at the PCI domains of EF3. is bound at the solvent interface, and the rest of the components in the subunit interface. Uh, Although both the complexes look very similar, but when we compare both of them, we see that the head of the ribosomal subunit moves up. So in the scanning conformation, we see that the head is moved up compared to the body. So if the body is more or less at the same position, whereas the head moves up. So if this is the closed conformation or the scanning arrested conformation, so head is positioned here with respect to body. In the open conformation, the body does not move, but the head is now moved up. So this actually allows, this widens the mRNA channel which is present between the head and the body. And we propose that this opening of this head would actually allow for the attachment or the recruitment of the mRNA in an open conformation. So here we can see the movement of the head, and from an open conformation, the head moves and comes close to the body. So in when the start codon is recognized. So the changes that we saw as, in between these two structures were that the tRNA moves closer to the body. So this is one. Then the EF1 again, it displaces from its original position and it's slightly displaced. So which would eventually then allow EF1 to leave the complex. The N-terminal tail, which I mentioned, which we saw why in the P-in state or when the tRNA was recognizing the start codon, was absent in in the complex, in the scanning complex. So N-terminal tail of EF1 gets or is stabilized only in a scanning arrested conformation when the start codon is recognized. In a scanning conformation, it is not, it is disordered. So the other thing what we observed was that the EF2 beta, which is shown here in red, interacts with EIF1 in a scanning conformation, and this interaction was not observed earlier. And we could show using genetics experiments as to that if you mutate the interface between EF1 and 2 beta, actually the scanning is affected. So this observation is real. And as the ribosome moves from scanning to scanning arrested conformation, we'll see that this EF2 beta moves away from the ribosome. So this shows you will see the closing of the head. So the head moves from open to closed position, and at the same time EF2 beta moves away from EF1. So here we see the conformational, the major conformational changes between in the ribosome while it is scanning to a scanning arrested conformation. So essentially EF3 seems to encircle the complete 40S in these complexes. So if the EF3 is shown here in gray, so if you the linkers here are missing. So if we imagine linkers connecting these dots, these gray points, then it seems to encircle the complete 40S. So we have further now solved higher resolution structures where we are able to now identify the regions of EF3 which is occupying the subunit interface, and this is actually EF3 beta. And we also found that EF3I, which is another subunit of EF3, relocates together with EF3 beta to the subunit interface. This shows just the fitting of EF3 beta into the density, and this is just to show that EF3I, which is again a similar protein but a smaller in size, will not fit. We did mutational analysis of different residues of EF3 beta to show that yes, this position on the ribosome is physiological, and it is these perturbations of these interactions affects the translation initiation. This paper is now on bioRxiv. So what we saw was that 3B and 3I, they actually relocate from the position on the from the back of the ribosome to the site of action, where in the subunit interface, where it can actually now contact EF1 and EF2. Now, according to biochemistry, it was known that EF3 can actually interact with EF1 and EF2, but it was not clear in terms of structure how it will bind, because In the previous structures, what was observed was that the EF3 binds at the solvent interface of the ribosome, whereas all the other components bind at the subunit interface. So it was not clear how a factor which is binding at the subunit interface would interact with factors which bind in the subunit interface. But now that with the relocation of 3B and 3I, or portions of EF3, we now can explain how EF3 interacts with both EF2 here, so EF3, 3B interacting with 2 gamma, and also with EF1, where we see regions of EF3C in contact with EF1. So this relocation explains how EF3 can actually coordinate with all the initiation factors to modulate each and every step of initiation. So we are looking at this open conformation of this ribosome and trying to explain how this open conformation of the ribosome during its scanning selects the start codon. And we have, in collaboration with Professor Prabal Maiti, who is also a co-organizer of this conference, we have looked at this particular problem. And this paper is in bioarchive. So what we have done here is we have mutated the start codon with different codons and tried to check at the energy penalty of doing so. This what we observed was that out of all the possible codons, namely 4 codons or 4 near-cognate codons are the ones which is having a very low energy penalty. The rest will have a high energy penalty. So in yes, the open conformation or the scanning conformation of the ribosome can actually discriminate between codons, and only it will move towards a clean state or a closed state only at start codon or the 4 different or 4 other near cognate codons. So what we have learned from these structures is that one of the first step is the formation of 40S11A EF3, where we see 1 and 1A bound at the A and the P site. EF3 is bound at the solvent interface. The next tRNA is brought in by EF2 complex, and we see a rotation of the head. When the mRNA binds to the ribosome, we see that the head is now within an open conformation, so that the mRNA channel is widened, and we see a relocation of EF3 to the subunit interface. When the start codon is recognized, we see the head moving down. The the end-terminal tail of EF1 is now ordered, and it approaches the codon-anticodon interactions, and we see the tRNA, the EF3 at the subunit interface. Sean, do I have time, or shall I stop here right now?
Maybe you can take a few more minutes because there was a question in between. So
yeah. Okay, sure. So we have observed till now. So the thing is that we didn't see in the complex is where is EF5. Now EF5 I mentioned is a GTPase activating protein, which activates the GTPase activity of EF2. And it is again it has 2 domains, an C-terminal domain, and it contacts EF1. So these biochemistry was known, but we were unable to see EF3 and EF5 in the complex. So we have now, some time back, we solved again a 48S where we were actually able to see EF5, only the N-terminal domain of it. So and if you see this color in cyan is actually the position of EF5. Now, if you notice from my previous slides that this was actually the position of EF1 in the previous complexes. So where we now see EF5 occupying exactly the same position. So just to show you the map, so this is the fitting of EF5 N-terminal domain in the map, and this is where how EF1 would look like. So it is definitely EF5, which is there in the map. I would like to mention that this, the N-terminal domain of 5 is actually a molecular mimic of EF1. So in some sense, this is mimicking EF1 at that position. So what we see is that once the start codon is recognized, so this is again comparing the structure of, this is EF1 bound to the tRNA at the P site, and this is EF5 N-terminal domain bound to the tRNA. So what exactly are we observing is that Once the start codon is recognized, EF1 is moved away from its original position and hence it leaves the complex. Once EF1 leaves the complex, the N-terminal domain of EF5, which is having a very similar domain as EF1, occupies that position. And here I am comparing the structure of the 48S where EF1 is there in orange, and in the green is the structure with EF5 and terminal domain. And you can see now in this particular position, the EF1 position will actually clash with the tRNA in this particular complex. So essentially what we are seeing is that once EF1 leaves the complex, the tRNA is further accommodated into the P site, such that it would have a steady clash with EF1 if it was there in that position. But in EF5, N-terminal domain now moves in to occupy that position so that EF1 cannot come back to this position, and EF5 is now bound at this, in this particular orientation. This is just to show that the arginine 15, which is essential for its GTPase activity of EF2, is further away from GTPase site of action in this particular conformation. This is just to reiterate this. This is in this conformation, it is not performing its actual function of gap activity, but in this function, in this particular conformation, it is actually playing a role in keeping EF1 out of the complex once the start codon is recognized. And we did a mutation analysis along with the collaborators' help to actually show that yes, mutations in regions of EF5 now will affect translation and the start codon recognition.
If
yes.
you can wrap up in another minute, then we'll have about 10 minutes for questions.
Sure. So in this particular structure, what we noticed that the EF3 domain that we had earlier seen in the subunit interface is now moved back. So what we are seeing is that the EF3 domains that were positioned here is now again positioned at the back of the ribosome. So they have moved back to their original position. So I had summarized till this point as to what was going on. So what we see next is that once EF1 leaves the complex, EF3, again, these regions of EF3 moves back to its original position. And now this position is now occupied by the N-terminal domain of EF5. So just to briefly mention that we are now looking at the translational control, which means how this process of translation initiation is actually regulated. And just to briefly mention that there are two steps at which the translation can be regulated. One is the tRNA delivery step, and this by regulating this step, the delivery of the tRNA to the ribosome is blocked. The next major mode of regulation is the mRNA delivery step, where protein like 4E binding proteins or 4G binding proteins can actually block the recruitment of the tRNA, the mRNA to the ribosomes. We have summarized these role of EF4 and these regulations in a perspective that we have published this year. So I'll finish at this point. So the take-home message is that all along the translation initiation pathway, we see large conformational changes in the ribosomal assembly, and this guides the ribosomal pre-initiation complex along the initiation pathway. So this is the pictures of the Cryo-EM facility at IISc, and I would like to thank IISc for the startup, the DBT Welcome Indian Alliance Fellowship, Jose, with whom I'm collaborating now and he was also my postdoc colleague at Venki's lab. So Venki's lab, for some of the work that I showed earlier, was done in Venki's lab. The collaborator Alan Hennebush at NIH, and I didn't mention in detail the computational work that we have done with Prabal, Professor Prabal Maiti and Ipsita and Biswajit from Prabal's lab. And thank you for your attention.
Thank you very much, Tanweer. That was a really lovely talk. So if there are questions, we have about 10 minutes for questions. So please go ahead.
Can I begin with my questions?
Please go ahead.
This is Debashish. Tanweer, thanks for a nice talk. Yes. I have a comment first and then a question.
Yes.
My comment is that you said that the molecular machine The concept was introduced by Bruce Alberts. Actually, historically, it was introduced by Marcello Malpighi.
Okay.
So he was a microscopist anatomist. So in 17th century, he introduced that. And since then, that term has been used extensively,
Okay.
particularly by biochemists in the context of muscle contraction, etc.
Okay.
So,
Thank you. Thank you for that.
Bruce Alberts popularized it. Yeah.
Okay.
So
Okay. Okay. So he
that's my
popularized
comment.
it. Okay. Thank you so
Yeah.
much.
Okay.
Thank you.
Now, my question is that as far as I know, in the translation initiation, first
Yes.
it is the small subunit which scans and finds the start codon site,
Yes.
and then the large subunit comes and binds with the help of various factors, right? But
Yes.
you mentioned that ribosome scans, so I was a little confused. Is this special case or
No, no. So ribosome means the small subunit of the ribosome scans the UTR, the untranslated region.
yeah, so it's the same that I have learned from textbooks, that it is the small subunit which scans it, then of
Yes,
course the
yes.
whole thing assembles at the
Yes. It is a small subunit which scans, and then once the start codon recognized, then only we see the assembly of the full ribosome.
Okay, so there another question of which I could never find any answer. In case you know the answer, that is, see, it's usually written that the small subunit is scanning, and scanning is directional.
Yes.
So the question is that if it's a directional movement, there must be supply of energy without which it cannot move on the average
Yes.
directionally.
Yes.
And so from where does this energy come?
Okay. So first of all, number one, we do not know completely the answers of these questions. So that is number one. When it is said that it is directional, I think it is overall directional. This is what it is meant. It is meant that it scans from 5' to 3'. Whether there is some backstep or anything in between, I think those details are not known. One of the question, next question is about the energy for scanning. Now, this is something which is again not known, but at least in eukaryotic initiation with EF4, there's a component called EF4A, which is a helicase, and it uses ATP for its helicase activity. So whether that provides the energy for it is yet to be figured out in detail. So because in an unstructured mRNA, so for example, in our structures that we used an unstructured mRNA, which also has an UTR region, so so the ribosome would have to scan for it, but we were able to do it without EF4 complex. So hence, in these scenario, it may have been just a diffusion, or somehow the ribosome is able to move on the mRNA. We are not clear, so we are not sure of that. But these are unsolved questions in the field.
So this helicase is associated with the small subunit?
Yes, so this helicase is a component of EF4 complex, and which actually brings in the mRNA to the ribosomal PIC. So and hence it will be attached to this complex when the mRNA is recruited. And it is known that these helices, 4A kind of leaves and binds again to these complexes. So multiple ATPs cycles will be required. So whether that will provide or that provides energy for the scanning of in cells is not very known.
Okay, thank you.
Yes. Yeah, but these are unsolved questions in the field.
Any other questions? Please go ahead, unmute yourself and ask, Tanweer. Tanweer, I had a question,
Yes.
might be kind of naive, but we sort of think a lot about, there's this question of how do you scan to find the start site, right? There's this question of 1D scanning versus combination of 1D, 3D search and so on. Is there
Yes.
something known in the ribosome? Like how exactly is this? And so even a simpler question before that is probably, on average, do we know how many nucleotides have to be scanned before the start site is found?
So on an average, the untranslated region in an yeast genes range in the range of about 50 to 60 nucleotides.
I see.
And in I think in humans it goes up to 100 or something.
I see. So that's not too much.
So that's not too much, but the thing is, in certain genes, for example, in humans, I think the number, the one of the highest ones is in the range of thousands. So it could be, for certain genes, it could be thousands of nucleotides before you actually reach the start codon.
I see.
And then the other complexity comes in when you have the AUG but in a wrong context, or you not in the presence of Kozak sequences. Then again, those AUGs are not really efficient start codons. So so uh,
So then is the search, do you think, is completely along that RNA strand, or do you think there could be some 3D diffusion as well? And if so, how would you even measure this in an experiment? Like, is this a purely...
Yeah, so the thing is, once the mRNA is bound, it kind of, the mRNA channel is will be almost like a tunnel, because then there are latches which closes in. So this is where the concept of open and closed conformation of the ribosome comes into picture. That in a closed conformation, the mRNA channel is almost like a tunnel. So if the mRNA is already bound, there's only, it can either go forward or backward. 3D kind of diffusion is unlikely. So it has to, and people have shown by experiment that one can, the ribosome actually looks at each and every point. So
interesting, I see. So most
so it is direction, one by one scanning of each and every nucleotide.
Right, I see.
And since as you see that the codons is pair of three nucleotides. So if we are scanning this particular say codon for at one point, the next step is not a jump of 3, but again a jump of just 1, because the next could be the AUG codon.
Right, absolutely.
So it is looking at each and every nucleotide during scanning. So this is where we try to answer this in some sense using this computational study, where we saw that in an open conformation, if we put AUG as a reference and we calculated the energy, and now if we mutate this AUG at, say, with one single point mutation at A or U or G and things like that, we saw that at if we mutate all the three, obviously we see a big energy penalty. If we mutate any two positions, we see again a big energy penalty. Now, when it comes to just single point mutations, we saw that only for 4 different mutations that the energy penalty is low. So perhaps while scanning itself, it can figure out whether this codon is just in terms of its base pairing, whether it has to, now the ribosome should shift to the next step of scanning arrested state, or it should continue scanning.
Right.
So but the details of how exactly it is done, and where the energy comes from, or it is just based on the movement of the head, because the ribosome, the head is free to move along with respect to the ribosomes, with respect to the body. Whether that gives some energy to it is not known.
Okay, great, thanks.
So
I see that Prabal has his hand up, so go ahead and please unmute yourself and ask the question. Maybe this will be our last question, because we are sort of running out of time.
Hi Tanweer, nice talk. Yeah, I just have a quick comment. This in the scanning arrested tag, when you are telling that this EF1 is leaving the complex.
Yes.
So then does it leave? I mean, but again, it has to assemble again, correct? So
no, so in the scanning arrested, so if you are talking whether it comes back to the scanning state.
Correct.
Okay.
Because again, it has to help it again, no?
Yes. So this is where if you see in our structure that we have solved, we saw that it moves away from its original binding side, but it is slightly shifted. Okay. So at this point, we still feel that there's a probability that it can go back to its original position if the ribosome now shifts back to the open state. Then the steric clash from the tRNA is gone, so it can go back to its original state. But if the tRNA remains in that scanning arrested state, then EF1 can no longer go back to its original position, and hence it leaves. So in the context of only start codon, which is AUG, it will leave the complex. In a near cognate state, like for the other group had shown when they did the computational work on the closed complex, they saw that the energy penalty of any other codon other than AUG is high. So hence, only AUG will be selected. So that it is kind of fine-tuned for AUG in the scanning arrested state. But for a scanning confirmation, what we saw that it may not be only for AUG, but also for some other mutations, some other codons it may allow. And that may be, this whole strategy of keeping it open to not making it specific to AUG, but to some other near cognate codons also may also allow for a higher speed of scanning. If the scanning was too tight and maybe rigid, specific for AUG, perhaps the scanning rate would have been affected. So
okay,
just to keep it coarse, maybe it allows for a speeder way of scanning it. And then there's another step where if there's a issue, then those near cognates can be checked it at scanning arrested state. So I think that is what is happening. This is what I feel.
Okay, fine. We'll discuss. Yeah. Okay.
Okay. Thanks a lot, Tanweer, for that lovely talk.